Hi, my name is Sahib, and I'm going to be presenting this paper for my biocancer presentation. Exosomes derived from HCC cells induce sorafenib resistance in hepatocellular carcinoma, both in vivo and in vitro. HCC over here stands for hepatocellular carcinoma, and so this paper will basically be investigating liver cancer, and HCC is the fifth most common cancer and the second leading cause of cancer-related deaths worldwide, resulting in 700,000 deaths annually. So this is obviously a very dangerous cancer and highly warranting of investigation and and research. Because anything relating to resistance against the drug treatment would result in lower survival rates. In this case, we're investigating sorafenib, and sorafenib is important because sorafenib, this article mentioned, is currently the only medication approved for the treatment of liver cancer. So anything, in this case, the exosomes, which are lessening its effect, would be really um, would be important to investigate and to reduce this resistance in order to get higher drug efficacy. So this paper basically it shows how these exosomes, these cancerous cell exosomes, induce sorafenib resistance in vitro by activating the HGF-c-Met AKT signaling pathway, which we learned about it, and inhibiting sorafenib-induced apoptosis. So just a quick background on exosomes. Exosomes are vesicles inside of, they're present in our cell-derived vesicles. They're in many eukaryotic fluids, blood and urine, and on culture medium of cell cultures. And this article mentioned them as playing an important role in the exchange of biological information and as substance transport carriers. So they're kind of like communication vesicles a little bit. So, and previous papers actually showed how, um, over here, previous studies suggested that these cancer exosomes resulted in enhanced AKT and ERK signaling, which and enhanced tumor cell invasion. And these two pathways are involved in, cancer proliferation when they're activated, when they're highly activated. So yeah, and we'll be investigating how uh, the exosomes inhibited sorafenib-induced apoptosis by activation of the HGF-c-Met AKT signaling pathways. So it's important to note before we start the first figure that the exosomes were derived from two different cell lines, MHCC97L and MHCC97H, and they're actually both invasive cell lines, highly invasive cell lines, cancerous cell lines, and you see hepatocellular carcinoma, HCC, right here. For simplification, I'll be referring to them as 97L and 97H. And there's also another cell line from which exosomes were taken, the LO2, or normal liver cell line. So these are regular exosomes that weren't inside cancer cells. So the first figure is an apoptosis assay. And for each assay, there was 10,000 cells analyzed, so each of these contains 10,000 cells. So an apoptosis assay shows the amount of cells in apoptosis. So each one of these little dots is a cell, and depending on which quadrant it's in, we'll know if it's apoptotic or not. So we've done this in class before, but how this assay measures apoptosis is through annexin 5 binding, which is shown on the x-axis. So cells release phosphatidylserine when they're apoptizing or when they're apoptotic, and annexin 5 binds to phosphatidylserine. So this x-axis right here shows how much annexin 5 is bound. So left to right indicates higher annexin 5 binding. So in these quadrants right here, there's high annexin 5 binding, and that's why these quadrants are when the cell is apoptotic. So, and on the y-axis, it's propydium iodine binding. Propydium iodine binds to DNA, and when the cell is late apoptotic, the DNA is outside the cell, and propydium iodine binds. So, high propydium iodine binding shows the late apoptotic cell in this quadrant right here. This quadrant is for necrotic cells. We won't be looking that much into it. So the first figure shows that uh, a control. So basically, it's no drug was added, and it's just a normal amount of cells. And you can see most of them are in the living 91%. They're just all living cells right here. Low Annexin-5 binding, low propydium iodine binding, just living cells right here. As we go on to the next one, sorafenib. So this is the drug we were talking about. So this drug obviously shows that it induces apoptosis in these cells because there's a much higher rate of apoptosis. See, 45% and 15.4% compared to this one right here. So sorafenib, a bunch of apoptotic cells. But then as we go on to sorafenib plus the regular exosomes, these are non-cancerous exosomes. Um, we can see that that the apoptosis was slightly reduced, about 6% in each of these two quadrants, but not that much overall. But then when we go to the next one, this is sorafenib plus the 97L exosomes. Now remember, this exosome was derived from cancer cells. So we can see over here, there's a much bigger decrease in late apoptosis. It was 45% or 46% right here. Now it's 36%, a 10% decrease in late apoptotic cells. Not much in early apoptotic, but there's a significant difference. So that kind of indicates that these exosomes, these cancerous exosomes, played a role in reducing the amount of apoptosis induced by sorafenib, the drug. As we go on to the next figure right here, we can see that there's an even bigger decrease in apoptosis because of the presence of the 97H exosomes. See, there's 25% right here, and there's 46% right here. So a huge decrease, and we can see all of the information in the graph too. There's a huge decrease in apoptosis rate, especially in the 97L and 97H. We can see, and these are the two cancerous cell exosomes, basically. But we can see compared to 97L, 97H showed a much bigger decrease in apoptosis. And that's, so it mentions this in the article right here. The apoptotic rate in the sorafenib 97H was significantly higher. This is because this cell line is more invasive than the 97L cell line. So the exosomes resulted in a much bigger decrease in apoptosis. And we find out how these exosomes led to a decrease in apoptosis through the next figure. So sorafenib induced apoptosis. The exosomes, basically, the cancerous cell exosomes, basically reduced it. And we'll see how over here. So these exosomes, HCC cell-derived exosomes, they affect sorafenib resistance via the HGF, c-Met, and AKT pathway. So as we go down over here, we can see this figure. But before that, we'll investigate the pathway they were talking about. So HGF, c-Met, AKT pathway. So c-Met is basically an HGF receptor, HGFR. And HGF is hepatocyte growth factor. Hepatocytes meaning liver cells. So yeah, we can see right here. So in the pathway, we can see over here, the receptor, which is a tyrosine kinase, gets phosphorylated when it binds to HGF through dimerization, then transphosphorylation. But the phosphorylated MET binds to PI3 kinase through the P85 region, as we can see over here. So PI3 kinase, when it gets activated, what it does is it goes and converts PIP2 to PIP3. So it adds a phosphate group. And what PIP3 does over here, since it's a kind of, what PIP3 does is it allows PDK, the protein-dependent kinase, to phosphorylate PKB, or protein kinase B, which is AKT. And when AKT is phosphorylated, it goes active and it phosphorylates a whole bunch of other proteins, and mostly it's a cell growth pathway. So and all the genes relating to those are oncogenes. So this helps the cell grow and avoid apoptosis, and that's how the exosomes basically reduce the amount of apoptosis. So as we can see in the graph over here in the first one, this is basically just the amount of HGF in the sample, HGF, hepatocyte growth factor. So basically the important part to note is we can see over here, there's much higher amounts of HGF when we add the 97L and the 97H exosomes. So that means this HGF c-Met AKT pathway is more activated. Um, so yeah, and then when we go on to the next figure over here, the main changes we see are in phosphorylated MET, phosphorylated AKT, and phosphorylated VEGFR2. So when we add the 97H exosomes plus sorafenib, there's a much higher level. There's a much higher level of phosphorylated MET when compared to just sorafenib. So this means that these exosomes, the 97H exosomes, they're activating this phosphorylated MET, and so they're activating the pathway, which leads to phosphorylated AKT, which leads to cell growth, cell division. So and this is better seen in the phosphorylated AKT compared to sorafenib right here. These two blots over here are much bigger, and those show a higher concentration of phosphorylated AKT in cells with these cancer exosomes. So they lead to more and more production of cells and less apoptosis. So, and we can see less in the sorafenib lane. Although there's not much difference in the phosphorylated MET for 97L exosomes, we can see the difference in the 97H, so that counteracts it a bit. And then in the phosphorylated VEGFR2, so this is vascular endothelial growth factor receptor. They play a role in angiogenesis and metastasis of cancers. So we can see when in the presence of sorafenib, it's less, but these exosomes increase the concentration. Even in the presence of sorafenib, they increase the concentration of the phosphorylated vector. And again, remember, phosphorylated means active. So we're not looking at regular AKT and regular c-Met. We're looking at the phosphorylated version. So which one is more activated? And in this case, in the presence of exosomes, phosphorylated MET or the receptor and phosphorylated AKT and phosphorylated VEGFR2 are much more phosphorylated in the presence of those exosomes and active. So in this next graph right here, so this Western blot is basically showing the action of crizotinib, which is a phosphorylated MET inhibitor. And as we said again, so phosphorylated MET activates the pathway, right? We can see over here there's much less phosphorylated MET when you add the crizotinib. So less phosphorylated MET, the pathway is less activated. So that basically means even in the presence of sorafenib, that basically means the crizotinib is helping the sorafenib, and there's less activation of this pathway. So that would mean more apoptotic cells, and the cancer wouldn't be helped basically. Although we can't see much of a difference in 97L, there is a lot of difference in 97H, as we can see over here. And we can see there's much less phosphorylated AKT as a result of the phosphorylated MET being less. And over here we have MK2206, which is a phosphorylated AKT inhibitor. And as we can see over here, so when you add the 97L exosomes, a good amount of phosphorylation, but there's less when you compare it to added MK2206. And remember, in both of these, there's already sorafenib in all of them. So that means that these drugs actually can help the action of sorafenib in cancer cells because they're reducing the phosphorylated AKT, and when this pathway is inhibited, that would help in reducing cancer. When it would help in reducing cancer because this is a cell growth pathway. So, and we can see it for the 97H too, less phosphorylated AKT when compared to regular 97H exosomes. So these two drugs might help in reducing the effect that the exosomes have on sorafenib. So that's an important find for scientists to help this drug, help sorafenib in treating liver cancer. So that's basically what this paper was about, how these cancer exosomes inhibit or induce resistance against sorafenib in these cells, in cancer cells.